Good morning, everyone.
Good
Good
morning,
morning,
sir.
sir.
Good
Morning,
morning,
sir.
sir.
Okay, so I think last time we had started with metabolism of amino acids, wherein we said that there are three reactions that we will be studying: deamination, decarboxylation, and transamination. Out of which we started with deamination, and we covered quite a few number of enzymes which are responsible for deamination. There are three more reactions under deamination, which we will discuss before we switch to decarboxylation. So let us cover that part first before we actually get into the decarboxylation part. So the type that we are going to discuss here right now is hydrolytic deamination. So as the name suggests, hydrolytic deamination, this is going to be a process which is carried out in presence of water. So we will do this with the general structure of amino acid and see what happens when deamination is carried out in presence of water. So what you basically do in this process is that you are going to get rid of this, and a hydrogen is added to the amino group from water such that it's given out in the form of ammonia, and the OH group of water is substituted over here. So what happens is that your structure looks like this, which is nothing but a hydroxy acid. So in presence of water, you are removing the amino group in the form of ammonia, and you are converting the amino acid to a hydroxy acid. So this is what we call as hydrolytic deamination. Similarly, you have a deamination which can take place as a reductive process, as a reduction process. We normally also refer to this as a reductive deamination. So this reductive deamination is the one which takes place in presence of water. So let us see how it is carried out. So again, I am writing a general structure of amino acid. This reaction will take place in presence of water, and there's going to be a reducing agent which will donate hydrogens. Okay, so that's going to vary from one reaction to the other as to what is going to act as the reducing agent. So that the basic job here is going to be to get rid of this. Okay, and obviously that's going to be released in the form of ammonia. Okay, so NH3 is going to be one of the products. I'm going to write it over here. Okay, NH3 is going to be one of the products in this particular process. So let us say that the hydrogen is basically coming from water. It's going to be added to the amino group, and you release it in the form of NH3. Okay, so that is one thing that you have to remember over here. The second aspect now is that you're left with OH, and you are left with these two hydrogens now, right? So what I am going to obtain over here is something like this, wherein I have substituted the NH2 group with a hydrogen. Okay, so what I am getting over here is a saturated acid. Already NH3 has been one of the products. Now, what am I left with? I used one hydrogen which is coming from these two H to substitute it over here. So now I'm left with one H and one OH. Okay, because यहाँ का एक हाइड्रोजन हमने अमाइनो ग्रुप के साथ अमोनिया रिलीज करने में यूज कर दिया, तो यहाँ पर एक OH बचा हुआ है और यहाँ का एक हाइड्रोजन NH2 के जगह पर अटैच किया तो एक हाइड्रोजन बचा हुआ है. So which means that a water molecule is going to be produced. Okay, so that's how in presence of water you're going to carry out reduction reaction and convert an amino acid to a saturated acid. I'm repeating once again. You remove the amino group hydrogen from water combined with the amino group to release it in the form of ammonia. So after loss of hydrogen in water, what are you left with? The OH group. And now you have two hydrogens. Out of these two hydrogens, one hydrogen is being substituted in place of the amino group to give you saturated acid. Okay, so now you're left with an OH group and a hydrogen, which means that together they are going to form one water molecule. So you form saturated acid, water molecule, and ammonia in the process. There is an another process of reduction also, which we need to understand, wherein it combines reduction, deamination, and decarboxylation together. Combines all three processes together. So let us see what happens in this. So again, a general amino acid is what we are writing over here. Okay, water molecule and two hydrogens. So in this process, as decarboxylation is taking place, this is going to be released separately. Okay, so CO2. This will be removed, a hydrogen will go over here and will be released in the form of ammonia, that is NH3. Okay, so what I will be left with is RC. I will substitute the amino group with a hydrogen and यहाँ का H तो as it is है. Okay, so what I am getting over here is a hydrocarbon. Plus OH is left, one hydrogen is left, so water. So a hydrocarbon is formed along with carbon dioxide, ammonia and water when you are combining reduction, deamination and decarboxylation. Okay, there is an another reduction also with which is combined obviously with you cannot say actually combined with deamination, but you will see that there is amine formation which takes place over here along with reduction. So let us also see that this is not exactly a deamination reaction, but you will see how microorganisms can carry this reaction out to form an amine. So this is your general amino acid structure. Okay, you're going to carry out reduction, so there will be a reducing agent which donates hydrogens, and you will see that what is going to happen is this particular part is going to be separated out. So you will get NH2H. Again, here we will substitute the entire COOH with an H, and this COOH will combine with another H to form formic acid. So you are releasing this entire carboxyl group in the form of formic acid. You're just combining it with an H which comes from the reducing agent and substituting that carboxylic group also with H and thereby forming an amine. Okay, so this is also a reaction. This is not deamination, but where reduction is involved. Are these reactions clear to you? Reduction, hydrolytic deamination, and combining reduction, deamination, and decarboxylation. Everyone?
Yes, sir.
Okay.
Yes, sir.
Yes,
So
sir.
write these two reactions down. Give heading as hydrolytic deamination. I'll gradually scroll the screen for you. Are you all done?
Yes, sir.
Yes, sir.
Yes.
Great. So let us go to the next set of reactions in metabolism of amino acids, which is basically on decarboxylation. You very well know that decarboxylation deals with removal of carbon dioxide. So the enzymes which work over here, we call them as amino acid decarboxylases. Okay, these are the enzymes which work in case of decarboxylation. Now, let me show you how the general reaction would basically look like. So you have your amino acid in the form of zwitterion. You're going to get rid of this. So it's substituted by a hydrogen from the ammonium group, and the carboxyl group removed is released in the form of carbon dioxide. Now, please understand that the enzyme amino acid decarboxylases, these are widely distributed for a basic reason that production of amine is a very important reaction which takes place in any living organism. Amines are one of the basic intermediates, precursors, which are needed for several anabolic reactions, especially when you are carrying out biosynthesis process for any nitrogenous compounds, amines may be needed. Other than that, amines also have a lot of physiological effects, like maintaining pH in the system is one of those. So necessarily decarboxylation of amino acid is not just a catabolic process. It may be brought about because we need amines for some biosynthetic process or to bring about some physiological change. We actually need it. Or, amines could also be precursors for some very important chemical components of our system, which we'll take examples of when we go ahead. To begin with, let us first talk about glutamate. Okay, we let us see what happens when glutamate is decarboxylated. The enzyme which works in this scenario, it's called glutamic decarboxylase. So let us see what happens in this scene. So this is L-glutamate. The enzyme which works on this is called as glutamic decarboxylase. You will see that this is the COO group that is removed and one hydrogen will be transferred over there as a substitute. So the structure that I have after the group is removed or it is eliminated in the form of CO2 looks like this. This is called gamma amino butyrate. Plus obviously I'll write the CO2 which has been removed. Now this gamma amino butyrate, it is a neurotransmitter which is found basically in the brain. So this clearly tells us that glutamate is not being decarboxylated in the human body for catabolic purposes, but to generate a neurotransmitter, right? It's very important to understand this. Similarly, histidine, which is again an amino acid, when decarboxylated, you will see that it gives rise to a very important component which you would have heard of quite often when you did your immunology and medical, is histamine, which is a vasodilator responsible for constriction and relaxation of your blood vessels. The process is very significant, and histamine is nothing but a decarboxylated product of histidine. Similarly, if you decarboxylate 3,4 dihydroxy phenylalanine, you get a very significant compound. Obviously, this is what we call as DOPA, dihydroxyphenylalanine. And what it gives rise to is dopamine, a very, very important intermediate again, because this intermediate is then utilized to form epinephrine, which you also call as adrenaline. Okay, dopamine is again not just important for synthesis of epinephrine, but dopamine by itself has a lot of significant effects, right from your learning abilities to sensations to some visceral processes. Dopamine has a very significant role to play. Similarly, if you carry out decarboxylation of 5 hydroxy tryptophan, if you decarboxylate this, what you get is another very important neurohormonal compound, which is called serotonin. Serotonin in humans is extremely significant for maintaining metabolic balance, because it controls all the metabolic activities which takes place in your system. So decarboxylation of hydroxytryptophan is responsible for this. Now, we have to understand that this process is not just taking place in animals or in humans, but it is also seen in plants, and you will also see it in microorganisms. Okay, most of the physiologically significant amines, they are produced by decarboxylation of aromatic amino acid, and that to L amino acid. Okay, most of the aromatic amino acid decarboxylases, you will find them in mammalian tissues. But as I told you, that it's not just specific to mammalian tissues, it can also be seen in plants and other animals as well. Bacteria themselves are a very good source of amino acid decarboxylases. You have yourself studied and practically performed also few of these. Lysine decarboxylase is one such. Most of these enzymes are inducible. So please understand that until and unless you grow the bacteria in presence of that amino acid in the culture medium, they will not produce this particular enzyme. Some of these, or rather most of these decarboxylases, they also require pyridoxal phosphate as a coenzyme. So until and unless pyridoxal phosphate is provided in the medium, the reaction may not go ahead. In some cases, even the Schiff's base would be an intermediate in the process of decarboxylation. So it may not be a single step process. There may be an intermediate which is involved in that process. Okay. I hope this part is clear to all of you. Please start noting down the reaction. You can give heading as decarboxylation. The enzyme obviously is specific on the basis of the amino acid that you're decarboxylating. So generally the family is referred to as amino acid decarboxylases. Please start noting them. Okay, hoping that all of you have noted the thing down. Now, after discussing decarboxylation, the next aspect that we are going to discuss is again a very, very significant aspect. And the last thing that we are discussing in terms of catabolism or metabolism of amino acids, post which we go to the fermentation aspect of amino acids. So, what we are talking about right now is transamination. So, as the name suggests, transfer of amino group from an amino acid, which acts as the donor of amino group, to an acceptor compound is the process of transamination. So, it's transfer of amino group. And believe me that synthesis of many amino acids is brought about by this process of transamination only. Okay, wherein the donor is an amino acid while an acceptor is a keto acid. So the moment the keto acid accepts the amino group, it forms the corresponding amino acid. Okay, and the amino acid which donates the amino group, जिसमें से amino group निकल जाएगा, that in turn will get converted into a keto acid. Let us see this in the form of a general reaction, so that may make the concept a little clear for you. So in this case, this particular compound that you are seeing right now is an amino acid. This amino acid is going to act as a donor, wherein it's going to donate this amino group. Okay. The acceptor in this process is going to be another compound, which basically is acting as, or is a keto acid, which is acting as an acceptor of the amino group. So this is obviously a very much reversible process. Once this part is removed, the amino group part is removed. This may take place in presence of water. You will see that this compound which donates the amino group will get converted to its corresponding keto acid. Okay, while the one which accepted it, you will see that it gets converted into the corresponding amino acid. So this is how basically many of the amino acids are synthesized. Now, your question should be that who carries this reaction out? So the enzymes which are responsible for carrying this reaction out are called as aminotransferases. So depending on which amino acid they are working with, there will be a name. For example, alanine aminotransferase or glutamine aminotransferase, glutamic aminotransferase. Okay. Now let us take an example. Maybe a specific example helps us understand it much better. So let us talk about glutamic aminotransferase. So glutamic aminotransferase is, as the name suggests, working with glutamic acid, which is obviously going to act as the donor of amino group, and your alpha Kg, which is alpha ketogluteric acid. Okay. So basically, why alpha Kg? Because alpha Kg is going to be formed in this process. क्योंकि तुम अगर अल्फा के जी में अमाइनो ग्रुप ऐड करते हो तो दैट्स नथिंग बट glutamic acid. Okay, so let us see what happens in this process. So let us have a general acceptor compound over here, which is a keto acid. It's a general molecule which is going to act as acceptor, and I have glutamic acid. So glutamic acid is COOH, CnH2H, CH2, CH2, COOH. This is nothing but L-glutamate. This is a reversible process. The enzyme that works here is glutamic amino transferase. Okay. So what happens over here to the acceptor is that you get the corresponding amino acid synthesized, plus as you have got rid of, you get rid of this and you get rid of this, both of it goes to the keto acceptor. That's how you have an H and NH2, and the oxygen gets transferred instead to glutamate. So you will see what you get now is COOH, C double bond O. This double bond O has come from the keto acid there. CH2, CH2, COOH. So this is nothing but alpha Kg. So you have converted glutamic acid to alpha Kg in this process, wherein NH2 and hydrogen has been transported or transferred to the acceptor, and from the acceptor, the oxygen has come over here. So you have converted glutamic acid into alpha Kg. Okay. Similarly, you would see that if you are working with the alanine aminotransferase, alanine aminotransferase mein alanine will be the donor, and you will see that alanine is converted to pyruvic acid basically. Okay, so depending on which particular amino acid you are involving, the product that is formed is also going to be different. Please understand this. Okay. Now, this particular reaction is seen in all organisms, whether it be higher plants, whether it be animals, whether it be microorganisms. You will see all living organisms showing you transamination reaction, and you will also see that there are variety of aminotransferases depending on the amino acid that is going to be involved. That surely could be specific to the organism, that which transamination reaction does the organism carry out on a regular basis. So you have to understand that. And when you talk about glutamine and glutamic acid, you have to understand that glutamine, we have discussed already, is a carrier of ammonia. Okay, so all excess ammonia normally is combined with glutamic acid and stored in the form of glutamine. Okay, and whenever that ammonia is needed, glutamine can be converted back to glutamic acid also. So that balance has to be maintained inside the organism. Depending on the availability of the components, the organisms may behave slightly different. Okay, now please understand that when you talk about animals, animals have some limitations. Okay, animals, they cannot synthesize all amino acids. We know this very well, because of which in our diet also we talk about essential and non-essential amino acids, because there are only some amino acids which we can synthesize, while the rest, it has to come from the food that we consume. And that's the reason why we end up depending on plants for our or animals for our most of the amino acid requirements, because they may assimilate it, and those animals also which we consume, they may not be able to produce all amino acids. They may be depending on their diet for the amino acids. So ultimately the burden is coming on plants. So plants have the ability to synthesize most of the amino acids, and that's how our requirement for amino acids is also going to be fulfilled. Please keep this in mind. Okay, so somewhere or the other, we are totally dependent on plants for the reserve of amino acids that we have. Okay, so quickly note this reaction down. Give heading as transamination. You can first note down the general reaction and then the glutamic aminotransferase one. तो पहले general reaction जल्दी से लिख लो. Then I'll scroll down the screen for you to note the glutamic aminotransferase reaction. Are you all done?
Yes, sir.
Okay, so that marks the end of metabolism of amino acids. And let me tell you that the part of Deamination, it is from two books. One is Conan Stumpf, and the four reactions that we saw today is from Saleh, AJ Saleh. Okay, decarboxylation and transamination is again from Conan Stumpf. The scanned part for Conan Stumpf for the entire unit I have already uploaded. AJ Saleh, you have the ebook with you. Okay, it's in the same chapter. थोड़ा स्क्रॉल डाउन करोगे जिसमें प्रोटीएजेस वाला पार्ट है, उसी में नीचे आपको बहुत सारे डीएमाइनेशन के रिएक्शंस मिलेंगे, जिनमें से चार जो रिएक्शंस हमने आज डिस्कस किए हैं वो आपको करना है. Okay, I will be uploading this PDF as I've done for proteases in the classroom. I will be uploading this PDF also under the same section. As I told you, I'll be editing that section itself so that all PDFs are there at one place. I hope you all have checked your timetable for internal class test. Have you all seen it? I had sent the link also to you on the WhatsApp. Okay, so please start preparing for that. Okay, and another very important thing that I wanted to convey is that I have posted a journal completion log sheet on the classroom today morning. Okay, please, you can open that sheet and you will be able to see what you are expected to do. If you want, I can just show that part to you once. So this is how the journal completion log sheet looks like. Okay, so what you are expected to do is that once you have completed a particular experiment, these are tick boxes, you can just go and click over it. Okay, only once you have completed it, not otherwise. Okay, and tick only for yourself, not for others. Is that clear? So as and when experiment instructions are given, we will go on adding them over here, and you can enter your, once you have completed it, you can just click on these click boxes and we'll come to know the status of where you have reached in journal writing. Okay. Okay, so let's stop here for today. And I am not starting the fermentation part because in 5 minutes we'll not be able to do anything, as it's again a chemical reaction. So I'm not starting with that part today. We will start with that section in our next class. Okay, we will be meeting tomorrow in our regular lecture, as Unilever project is postponed for a few weeks due to the COVID scenario. So we will be meeting tomorrow in our regular lecture at 8:35. So we will discuss about fermentation of a single amino acid, and if time permits, we will get into fermentation of pair of amino acids. Okay, that is it for today. Thank you, and stay safe. See you all in the practicals today.
Thank you, sir.
Thank you, sir.
Thank you, sir.